everyone, today we'll talk about carbohydrates. Generally, carbohydrates are known as the hydrates of carbon, having the general formula CnH2O. They consist of mainly of carbon, hydrogen, and oxygen. Besides that, they may also contain nitrogen, sulfur, and phosphorus. Examples include glucose, lactose, sucrose, starch, cellulose, glycogen, galactose, fructose, starch, etc. Now, definition of carbohydrate is carbohydrates are either polyhydroxyaldehyde or ketone or compounds which produce them on hydrolysis. So let's try to understand this definition with the example. So this is the structure of glucose molecule, and as we can see there are multiple hydroxy. Hydroxy means the OH, that is the alcohol. So this is a polyhydroxy hydroxy, and if you see C1, this is an aldehyde group. So this is a polyhydroxy aldehyde. This is a polyhydroxyaldehyde. And this, and let's take another example of fructose. Here also we see multiple OH group. So this is polyhydroxy, and see this is a ketone group. C2 is the carbonyl carbon, so this is a ketone group. As we know from the organic chemistry, like let's say this is the carbonyl carbon and this carbonyl carbon. If there are two R groups attached, then this is a ketone group. As we can see in this is one R group, and the rest, this is another R group. So this is a ketone, and if to the carbonyl carbon, if one of the R group attached is H, then this is called as an aldehyde group. So this is an aldehyde. So this is a polyhydroxy ketone, and this is a polyhydroxyaldehyde. Now, carbohydrates, depending on the number of monomers present, they are classified as monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides, so mono means one, and saccharide means sugar. So the monosaccharide, they cannot be hydrolyzed further as they are composed of only one unit. And example include glucose, galactose, fructose, mannose, and either it could be an aldose sugar or a keto sugar, as we have already seen. Monosaccharides are further classified based on the number of carbon present. They're classified as triose, tetrose, pentose, hexose, and heptose. Hexose, as we have already seen, glucose and galactose, they contain glucose and fructose. They had the 6 carbon, so they are called as hexose. Triose having 3 carbons. Example include glyceraldehyde and DHA. DHA is dihydroxy dihydroxyacetone. Tetrose having 4 carbon, tetra means 4, so having 4 carbon, example is erythrose. Pentose means, penta means 5, having 5 carbon, example include ribose and the deoxyribose, the sugar present in the RNA and the DNA. Hexose include glucose, galactose, fructose, mannose. Hexa means 6, having 6 carbon. Heptose means having 7 carbon, example include glucose. Now let's see, try to understand the structure of these monosaccharides. Glyceraldehyde. So glyceraldehyde, it is an aldose sugar. So there is an aldehyde group at sequence. So this is an aldose sugar, and it has only 3 carbon. 1, 2, 3. So they glyceraldehyde and dihydroxyacetone. They are the smallest sugar present. possible. So this is an example of aldo triose. Aldo means having an aldehyde group, and triose means having the three carbon molecule. Similar, it is its DHA, dihydroxyacetone is its respective keto sugar. So it is also a triose sugar. There are three carbon, one, and there instead of the aldehyde, it is actually a ketone sugar. So this is a keto triose. Now let's take example of glucose and fructose. This we have already seen that there are six carbon, one, five, and six. So this is, and as we have seen, aldehyde group. So this is an aldohexose sugar, and its respective keto sugar is ketohexose. So this is also an hexose sugar. There are six carbon, one, five, and there is a ketone group over here instead of the aldehyde. So this is a ketohexose. Now next, this is ribose and deoxyribose. Ribose is present. It is present in RNA, and deoxyribose is present in DNA. So ribose, it is a pentose sugar. One, two, and one. So ribose is a pentose sugar, five carbon, and it has an aldehyde group. So it is an aldopentose. Now deoxyribose, it is also an aldopentose present DNA, and the difference between ribose and deoxyribose is it is 2-deoxy deribose. D means removed or lacking, oxy means oxygen. It means that the oxygen at the C2 has, it has been removed. So it is 2-deoxy deribose. And this is also an aldopentose. And it is present in DNA, and ribose it is present in RNA. Now oligosaccharides. Oligo means few. So they may contain monosaccharide. They may contain few monosaccharide. It could number from 2 to 10 or 20. And the two sugars are joined by the glycosidic. So here, what does it mean? It means that the two sugar molecules are joined by a glycosidic. We'll learn about details of this bond later. Now, most dependent of the among the oligosaccharides are the disaccharides. So, as the name suggests, di means two, and saccharide means sugar. Now, lactose. These are the common examples of disaccharides. So, lactose is milk sugar, which is present in the milk, and the constituent monosaccharides are galactose and glucose. Sucrose, which is the common table sugar we consume. So the constituent monosaccharides are glucose and the fructose. Maltose, it is usually present in seeds. So the constituent monosaccharides are two glucose molecules. It is a disaccharide of glucose. Now uh, polysaccharides. Polysaccharides, they may contain more than 20 monosaccharides. So they may contain up to hundreds or thousands of monosaccharides, and the different sugar moieties are joined by the glycoside. Now, example includes starch, which is present in rice, wheat, and it is starch is a polymer of D-glucose. So, which means that there are multiple units of D-glucose only. Now, cellulose that is present in plant cell wall, it is also a polymer of D-glucose. Glycogen, another example is glycogen. It is an animal storage carbohydrate. So, in animals, including human, the carbohydrate are stored in the form of glycogen. Okay, and it is a polymer of D-glucose. We'll learn about details of these disaccharides and polysaccharides later on. Let's try to just have an example of this starch. how does the polysaccharides look like? So starch, there are two components of starch, amylose and the amylopectin. Okay, so amylose is a linear component. There's no branching. Here we see this, there is one alpha 1,4 glycosidic linkage between two glucose molecules. So this is a linear component, and amylopectin is a branch component. So there is a branching. These are between these linear chain, between different linear chain, there is a cross linkage by alpha 1,6 glycosidic linkage. We learned about all these disaccharides and polysaccharides in detail later on. Now absolute configuration. So we have here, we have been coming across about D-glucose, it is a polymer of D-glucose, D-fructose, D-glyceraldehyde. So what is the difference between this L and D form? So L and D form. So in nature, this only D form of the sugar is present. D form of the carbohydrates is present. Now this is an absolute configuration. This is based on the structure of the glyceraldehyde. Now this is a glyceraldehyde molecule to look at. Now in the glyceraldehyde molecule, this C, central carbon, is the chiral carbon. This is a chiral carbon. This is a chiral carbon. And if we see over here, now this OH is on the right side. When this OH is on the right side, it is labeled as D, and when this OH is on the left hand side, then it is labeled as L. This L and D, it is an absolute configuration, and is it has no correlation with the L and D configuration, which represent the optical activity of the molecule. So a D molecule could be dextrorotatory or levorotatory. Similarly, an L molecule could be D or L. So it is irrespective. This is just for the absolute configuration of the molecule. Now, labeling all the carbohydrate molecules are labeled based on the structure of the glyceraldehyde molecule. Let's say, take this example. Now, in this, if we look at this is an example of an hexose. So, in uh, this fifth carbon. Okay, so let's first talk about the chiral carbon. In this, these C2, C3, C4, and C5. These are the chiral carbon molecules. Chiral carbon. Chiral carbon are in this molecule C2, C3, C4, and C5. Now, if we see C6, this is not a chiral carbon because there are two H. Now, C5, this is the highest chiral carbon present. So, and this OH is present on the left hand side. So, this is an L form of the molecule. And if this OH would have been present on the right hand side over here, then it would have been the D form. Okay. So it is based on this D and L configuration is based on the structure of glyceraldehyde molecule, and in this we have to take in consideration the highest chiral carbon of the molecule, and then we have to look for the position of H and OH. If the OH is on the right side, then it will be a D form, and if OH is on the left form, then it is a L. Now, what is a chiral carbon? So, this is a chiral center. This is a carbon molecule, and if the four group carbon has a valency of 4, so if the 4 R group attached to this carbon are different, if the 4 carbon molecule 4 R group attached to the carbon are different, then it is called as different, then it is a chiral carbon. But if any of them two are same, then it is not a chiral carbon. So in case of this, if we see this aldehyde molecule, there is a double bond present. So it can be represented as CH, this R group, and O. So these two are same. So it won't be a chiral carbon. Similarly for the ketone group, if there is a carbon in carbon, that won't be a chiral carbon. If we look at C6, this OH, this H, there are two H. So there are two same R group. So that's why it is also not a chiral carbon. If we look at the C2, in this is one R group, and this is another R group. This is third R3, and this is R4. So all four groups are different. That's why it is a chiral carbon. Now similarly, if you look at the C3, if you look at the C3, this is one R group for C3. This is another group. This is another R group, and this is the fourth R group. So all the four R group are different. That's why it is a chiral carbon. Similarly, you can take look for C5, right? C5. Okay. Now, I hope it is clear what is a chiral carbon, and you can identify if you are given a molecule, you should be able to identify what if the carbon is chiral or not. Now, so if a molecule is having this double bond or this any bond, triple bond, then that this carbon won't be a chiral carbon. If the molecule has this, if the carbon is involved in double bond or triple bond, then it won't be a chiral carbon. Now, this is this representation. This linear representation, is known as Fischer projection, whereas this cyclic presentation of the molecule is known as the Haworth projection. In the Haworth projection, this is an open chain. This Fischer projection, this is an open chain. This is a linear molecule, and this is known as open chain form. Whereas if you see this, this is known as closed. This is a ring structure, closed ring structure. This is a ring structure. Closed ring structure. In this ring structure, these molecules which lie on the ring, these represent plane of the paper, and these molecules which are on this perpendicular line, they represent groups above or below the plane. So, atom or molecule present on this over the perpendicular line, it represents atom present over the plane, above the plane, and those molecular atom which are represented below this perpendicular line, it represents molecule below the plane. So similarly here, it so in this 6C1, C2, this C3, C4, C5, and this 6 is the oxygen molecule. These 6 atoms lie in the plane, and this these molecule these molecule lie above the plane. Similarly, this molecule, this which no, no, these lie below the plane, and this see all this CH2H, it is out of the ring. The sixth carbon is out of the ring, and it is lying above the ring. So this is an Haworth projection, and this is an Fischer projection. Now, how this ring and the closed structure forms. So basically, when in aqueous solution, there is hardly any molecule which is present in the open form. Most of the sugar, when dissolved in water, they exist in the closed ring structure. So how this closed ring structure forms is actually by the formation of due to the hemiacetal ring formation. So this is an aldehyde. What is an hemiacetal reaction? When an aldehyde group reacts with an alcohol group, it forms an hemiacetal reaction. Okay, so this is the hemiacetal group. Now, in this molecule, this ring formation takes place due to an intramolecular hemiacetal ring formation. Here, intramolecular, intramolecular hemiacetal ring formation. Why it is called as intramolecular? Intramolecular means within the molecule. When the two reacting groups are part of the same molecule, then it is called as intramolecular reaction. So, sorry. In this uh, reaction, actually the aldehyde group and the OH group are part of the same molecule, molecule. That's why it is an intramolecular hemiacetal. Now, this aldehyde groups attacks the OH group on the C5 or the C4, or it could be C4 as well. So then this hemiacetal ring formation takes place, and the linear chain is converted to the cyclic. So it is in the solution. Most of the sugar they are present in the closed ring formation, and only very less percentage present in the linear form. Now pyranose and the furanose form. So this is a pyranose form. So it is when it is made up of 6 atoms. Out of these 6 atoms, 5 are carbon and 1 oxygen. So why it is called as pyranose? Because it represents a molecule pyran in the organic chemistry. So this is a pyranose form. This is the pyranose form where this is C1, C2, C3, right? C6 is out of ring. So C1 attacks the C5 of OH, C5 as OH of the C5, and that's why this pyranose ring. Furanose, furanose form. Why it is called as furanose form? Because it represents a molecule furan in the organic chemistry. So it composed of, it is a 5 atom rings. There are 5 atoms in the plane. The 5 atom ring, of which 4 are carbon and 1 oxygen. So you can see this. This is the pyranose form and this is the pyranose. Glucose, here it is shown in the pyranose form. It can exist in the pyranose form. Now, when okay, these are at the what are the different anomers? Now, when this is dissolved in when, let's say, glucose is dissolved in the water, so it can either take alpha form or the beta form. So what are these alpha or beta forms? These are actually called as the anomers. Now what are anomers? So here if we see this molecule, OH is above the plane, and here the OH is below the plane. This is the difference if you compare these two structures. So now when this, if you see this molecule, this OH and this CH2OH, they are on the same side of the plane. They are on the same side of the plane. they are on the same side of the, when they are on the same, then this OH group on the C1. C1 is the anomeric carbon because C1 takes part in the hemiasteral reaction. So C1, this C1, this C1 is the anomeric carbon. So this C1, when this OH on the C1 and the 6CH2OH, this is the sixth carbon. Okay, so when this OH and the sixth carbon they are on the same side of the plane, this is called as beta. This is known as beta configuration. And if you see this, CH2OH, this is the sixth carbon, and this OH on the C1. C1 is the, that is the anomeric carbon. If they here they are on the opposite side of the plane. This is above the plane. This OH is the below the plane. So they are on the opposite side of the plane. That's why this is called as an alpha configuration. This is the difference between the alpha and the beta, uh, beta glucose, and these are called as the anomers. When the two molecules differ on the configuration only at the C1, only on the anomeric carbon, then they are called as the anomers. Now we also come, when we study carbohydrate, we come across reducing and the non-reducing sugar. Now what is a reducing sugar and what is a non-reducing sugar? Now sugars which contain a free or potentially free reducing group are called as a reducing sugar. Example, all monosaccharides all monosaccharides are reducing sugar, and lactose, maltose, they are the reducing sugar. How? You see, this is the glucose. So, if you see glucose, they have this free aldehyde group, and the aldehyde, ketone, all these are strong reducing agent. So, this all monosaccharides they contain, all monosaccharides they contain either a free aldehyde or free ketone group. That's why uh, they, all monosaccharides are reducing sugar. Now like in case of disaccharide or other sugar, we'll see like why some disaccharides are reducing and why others are not. So this either they have this free aldehyde group or they have this potentially free. Potentially free means that this bond can, if it is present in a circular, in a ring, closed ring structure, then this bond can form, and there will be generation of this free aldehyde. So either they have this free aldehyde group or potentially free aldehyde group. That's why it is a reducing sugar. Now what is a reducing sugar? So reducing sugar or reducing molecule is something which can reduce something. So this aldehyde group, let's say what it can reduce this cupric ion to 2 plus ion. See here the oxidation state is 2 plus, and here the oxidation state of copper is plus 1. So it has, this aldehyde group has reduced this cupric ion to 2 plus ion. So this act as a reducing group in this molecule, and that's why this is called as a reducing sugar. And while doing this, when a molecule reduces, when it does the reduction of something, it itself gets oxidized. So this aldehyde group is oxidized to acid, carboxylic acid here. So an aldehyde group when oxidized, it forms an acid. So it is a carboxylic. Yeah. So this is the case of monosaccharides. So all monosaccharides they have either a free aldehyde or a free ketone group or a potentially free. If it is in a closed ring structure, this ring can open and there will be generation of the free aldehyde or free ketone group. That's why they have either, they will have this potentially free aldehyde or ketone. That's why all monosaccharides are reducing sugar. Now, in case of disaccharide, we look at the structure of maltose. In this uh, aldehyde group of this sugar is involved in the glycosidic. Okay, this is the maltose structure of maltose, and these are two glucose molecule. One, two. I have told, if you remember, maltose is a disaccharide of glucose, and the linkage between two molecules is alpha 1. Now these are two glucose molecules linked via a glycosidic bond. So in this glycosidic bond, C1, the carbon, this aldehyde group of of this first molecule is involved in the glycosidic. That's why it is not a that's why it is not available for reduction. And but if we see this C1 of the second molecule, this is it can generate the free aldehyde group. Okay, so this is here we could see potentially free reducing group. That's why it act as a reducing sugar. Now similarly we'll see in the structure of lactose there will also one free potentially free reducing group. But if we see at the structure of sucrose is also a disaccharide. It is made up of glucose and fructose. Now, if we see the C1 of the glucose, glucose is an aldohexose, and C1 is the aldehyde group. C1 is the aldehyde group. So this C1 is the aldehyde group. It is involved in the glycosidic bond. And in case of fructose is an aldoketose. It is a ketohexose. Ketohexose. And if you remember, the C2 was the carbonyl group. CH. So the C2 was the carbonyl carbon. And if you look over here, C2 is involved in the glycosidic. So C1 of the glucose, C1 of the glucose and C2 of the fructose are involved in this glycosidic. So there is no free reducing group available in the sucrose. That's why it is a non-reducing group. That's why it is a non-reducing sugar, because it does not have any free reducing group. Any free reducing It does not have any free reducing group. That's why it is a non-reducing sugar. That's why it is a non-reducing sugar. And why it does not have a free reducing group? Because C1 of glucose and C2 of fructose, both of which are which are reducing group, reducing group are involved in the glycosidic group. Are involved in the glycosidic bond formation. Okay, I hope it is clear. Now, if we look at the structure of the starch, we have already seen this structure. Now, I have told this that there's a linkage of alpha 1,4. Alpha 1,4 means that the C1 of one molecule is linked with the C4 of the another sugar here. So the C1, they all are linked. They are involved in the glycosidic bond. So there is no free aldehyde group. The free aldehyde group of every molecule is involved in the glycosidic bond. So there's in the thousands of molecules, there are very few groups that are available for reduction. So in the test tube, we'll hardly see any visible reduction because of these free ends. So that's why it is a non-reducing sugar, because here all in the case of polysaccharides, the free reducing group, the C1 will be involved in the glycosidic formation. So there will be hardly any free reducing group. In the thousands of the sugar molecule, there will be barely few molecules, few uh, every reducing group present, and in the total reaction, if we put it in the test tube, there will be hardly any reduction visible in the test tube. That's why it is a non-reducing sugar. Now we come to the basics. What is reduction actually? Okay, if you don't know, like if you want to revise it. So reduction or oxidation. Oxidation is loss of electron, and reduction is gain of oxidation is loss of electron. So if you look at this okay, we all know that we have all have studied like that. Okay, oxidation and reduction reaction, they never happen in isolation. The reaction is always coupled. So if we see this reaction, the formation of NaCl, Na and Cl. So the in here the in the native state, the oxidation state of sodium is 0, and here it is +1. So it has undergone oxidation. And if we take example of chlorine, here the oxidation state is 0, and here it is -1. So it means that it has undergone reduction. Now if we split this into half reaction, we can see that sodium has lost one electron. And oxidation means, the definition of oxidation is loss of electron. Loss of electron, oxidation. Oxidation is loss of electron. And reduction is gain of electron. So chlorine, it has gained one electron, and the oxidation state has become -1. So here sodium has undergone oxidation and chlorine has undergone reduction. So now if you see this, sodium has given this electron. Sodium has given this electron for the reduction of chlorine. Okay, that's why sodium has acted as a reducing agent. And chlorine, it has uptaken this electron. Chlorine has uptaken this electron. Okay, that's why it has acted as an oxidizing agent. Okay, so the one reducing agent, the one who act as a reducing agent, it itself get oxidized, and the one which act as an oxidizing agent, it itself gets. Now reduction of sugars. If we see oxidation, I have already told. Reduction, when aldehyde groups are reduced, when aldehyde groups are reduced, they form the alcohol. So this aldehyde group, when this molecule, this analdose molecule is reduced, it forms the corresponding aldetone. So when aldehyde group reduced, this aldehyde group is reduced to an alcohol group, and the aldose sugar will form the corresponding aldetone. For example, the glucose will form the glucidone. Now oxidation. Now oxidation of aldehyde, as I already told, oxidation of aldehyde forms the carboxylic acid. This we have seen. So, but in case of sugars or when we study the oxidation of monosaccharide, this okay. One more thing, oxidation of alcohol. When alcohol is oxidized, it forms aldehyde. Aldehyde. And this when this aldehyde are further oxidized, it forms the carboxylic acid. So there are multiple this here we have aldehyde group as well as this alcohol group which can undergo oxidation. Now depending upon the conditions present, these will undergo oxidation. Now because this aldehyde group, it is a strong reducing agent. It is a strong agent, so it can be oxidized under mild oxidizing condition. For example, under bromine water or sodium hypoiodide. So even in the presence of mild oxidizing agent, this aldehyde group will be oxidized to acid. Okay, in that case, it will be an aldose will form its respective aldonic acid. In case of glucose, it will be called as glyconic acid. Now, in case of a strong oxidizing agent, for example, like HNO3, H2SO4, both this aldehyde group and the alcohol group, C1 and the C6, they could be oxidized. So in that case, the C1 and the C both will be oxidized to COH, carboxylic. So that is called as an alderic acid. In case of glucose, it will be a glycolic acid. Now, in case if this CHO, the C first carbon is protected by some functional group, then this C6 will undergo oxidation to the carboxylic group in presence of the oxidizing agent, for example, dilutous here. So in that case, it will form a uronic acid, and in case of glucose, it is called as glycolic acid. These are like so. These are the different product of monosaccharide under different oxidation conditions. Thank you. If you have any doubt regarding any of these things, you can put it in comment and I'll try to answer. And for further, for see more lectures based on carbohydrate or other biochemistry lectures, please subscribe my channel and do put in comment like what on which topic do you want this discussion or the lecture. Thank you so much. Please subscribe my channel.